So today we're going to be going through our scientific method lessons. This is day 3 out of 5. Today we're going to be learning how scientists solve problems. So how do scientists solve problems? By utilizing the steps of the scientific method. So for our problem today we have, how does our heart rate change after exercise? Our observations. Our observations from prior experience tell us that heart rate tends to increase after we exercise, such as running or weightlifting. And we could get most of our research from libraries, scientific journals, and internet sources. Our hypothesis. If a person does exercise, let's say 30 jumping jacks, then his or her heart rate will increase. And see how we use the if and then words in our hypothesis. And to get even more specific, we can add the type of exercise, like we did here with jumping jacks, how long or how many, like how we did with the number 30, and the amount of change. Then this is our controlled experiment. Remember, an experiment is done to determine the validity of your hypothesis. We are going to design an experiment that will allow us to measure heart rate after we exercise. This is our controlled experiment. So the control group, it gets normal treatment, meaning no exercise and it's just the regular heart rate. Our experimental group gets the substance or the treatment you are testing. So in this situation, the experimental group would be doing the exercise. In our exercise experiment, which is the control group and which is the experimental group. Our procedure. Divide students into two groups. Students in one group will be instructed to complete 30 jumping jacks. The students in the other group will do no activity for the same time period. The first group is exercising. Have the students measure their heart rate for 1 minute during rest and write it down. Students from both groups will recheck their heart rate and compare it to their own initial heart rate. Compare the results from both groups to see if there is a difference between resting and exercise. And for our experiment, there are many different types of variables, which are factors that can change. We have the independent variable, which is what the scientist controls or changes, which in this situation is the exercise. We have the dependent variable, which is the variable that is observed that changes because of the independent variable, which is the heart rate. So the independent variable is a thing we can change, and the dependent variable is a thing that changes because of the independent variable. And then we have our controlled variable. Variables that are the same in each group, so in our control group and in our experimental group. So that would be age, exercise type, and time of day, etc. Constants, controls, and controlled variables. Both groups should be treated exactly the same except for the independent variable. Which is the one thing that is being tested. What are the constants in this experiment? We have age of participant, number of people in each group, and athletic conditioning. Identify the dependent and independent variable. In an experiment studying the effect of sleep on student achievement. So the dependent variable would be the effect of sleep, and the independent variable would be student achievement. And in an experiment studying the effect of caffeine on worker performance. So the dependent variable, which is the thing that changes, is the worker performance, and the independent variable is the effect of caffeine. So that would mean caffeine has an effect on worker performance. In an experiment studying plant growth in different colors of light, the dependent variable would be plant growth, and the independent variable, which is the thing that we can change, are the different colors of light. Sample sizes. The number of things included in each group. The bigger the sample size, the better the results or conclusion will be. How many people should we use? What is left to do? Conduct the experiment, collect data, analyze data and make a conclusion, refer back to our hypothesis, repeat the experiment to check the validity, peer review of work, alter procedure if necessary. And that's for our lesson today.